StemCells103, Frequently Asked Questions Part 3. This presentation was prepared by Dr. Max McLeod, D.O., N.D., Ph.D. Doc Max is also known as the Nutrition Ninja Doc. He's devoted over 5 decades to studying health and health optimization while being in clinical practice since 1979. He has 3 postgraduate degrees in the health sciences and is a contrarian, always looking for better ways to help people overcome their health challenges and achieve their health goals. He has been doing regenerative injection therapies since 1993 and has added human tissue products including stem cells, growth factors, collagens and cell signaling molecules over the past several years. As part of our continued series on stem cell frequently asked questions, we have taken some of the top questions from the top results of online searches. The website, the questions and answers will be cited, and Doc Max will provide comments and corrections as appropriate. First, we will cover a number of Q&As from Washington University Physicians website at the URL shown. Question number 1. What are stem cells and why do we hear so much about them? Stem cells help to create new cells in existing healthy tissues and may help repair tissues in areas that are injured or damaged. They are the basis for the specific cell types that make up each organ in the body. Stem cells are distinguished from other cells by a few important characteristics. They have the ability to self-renew. This is also true of most cell types. They have the ability to divide for a long period of time. This is also true of most cell types. And, under certain conditions, they can be induced to differentiate into specialized cells with distinct functions, phenotypes, including, but not limited to, cardiac cells, liver cells, fat cells, bone cells, cartilage cells, nerve cells, and connective tissue cells. This is their unique ability and varies by the type and source of stem cell. The ability of cells to differentiate into a variety of other cells is termed multipotency. What scientists learn about controlling stem cell differentiation can become the basis for new treatments of many serious diseases and injuries. Question number 2. What is the difference between embryonic and adult stem cells? Some organs contain stem cells, called adult stem cells, that persist throughout life and contribute to the maintenance and repair of those organs. Not every organ has been shown to contain these cells, and generally adult stem cells have restricted developmental potential, in that their capacity for proliferation is limited and they can give rise only to a few cell types. Embryonic stem cells, by contrast, can divide almost indefinitely and can give rise to every cell type in the body, suggesting that they may be the most versatile source of cells for research and transplantation therapy. Comment: the vast majority of stem cells are actually somewhat dormant cells called parasites. Parasites are mainly located on the outer surface of blood vessels throughout our bodies, and that includes every organ and gland. When there is an injury or damage to that area, the parasites are awakened from their dormant stage and activated. Once transformed into fully functional stem cells, they assess the damage and begin the process of repair by a combination of actions including directing other cells to perform various functions such as producing specialized proteins, activating additional stem cells, replicating, and differentiating into the cell type needed to carry out the repair. Question number 3. Where do stem cells come from? There are several sources of stem cells used in research. Embryonic stem cells are obtained from the inner cell mass of a blastocyst. The blastocyst is formed when the fertilized egg, or zygote, divides and forms 2 cells, then again to form 4, and so on until it becomes a hollow ball of about 150 cells. The ball of cells, now called the blastocyst, actually contains 2 types of cells, the trophoblast, and the inner cell mass. The inner cell mass contains the pluripotent stem cells that can be isolated and cultured. Stem cells are also found in differentiated tissues and organs throughout the body. Often referred to as adult stem cells, or tissue-specific cells, they have not been identified in all tissues and organs, but in many cases, they do exist and have a confirmed role in repairing and maintaining tissue that has been injured or damaged by disease. The adult stem cells can be isolated from samples of the tissue, with the cells suspended in liquid and separated based on cell surface markers using fluorescence-activated cell sorting, FACS. Blood from the umbilical cord of a newborn baby also contains blood stem cells and is often harvested and banked for future use, either for the benefit of research or for future treatments that the donor may require. The amniotic fluid is another rich source of stem cells that are multipotent and often more robust than stem cells derived by other means. Lastly, induced pluripotent stem cells, iPS cells, can be derived from the large pool of differentiated cells in the body, e.g. skin, fat, muscle, etc., which are transformed into an embryonic-like stem cell state. Comment: they left off the biggest, most convenient source of stem cells, afterbirth products including the umbilical cord and placenta. These stem cells are much more pluripotent than those derived from specific adult tissues. This makes them ideal for both research and treatment. Question number 4. What are induced pluripotent stem, iPS, cells? Induced pluripotent cells are derived from somatic, adult, non-germline, cells, which have been reverted to an embryonic stem cell-like state. Like embryonic stem cells, iPS cells can be differentiated into any cell in the body and are therefore considered pluripotent. The process of creating these cells, often referred to as reprogramming, involves introducing a combination of 3 to 4 genes for transcription factors delivered by retroviruses into the somatic cell. More recent methods have replaced and reduced the number of genes required for the transformation, used alternative delivery methods to get the genes into the cell, or sought to replace the genes with chemical factors. Cells can be taken from patients with specific diseases such as ALS, Parkinson's, or cardiovascular disease and induced to form iPS cells. Multiple uses can be derived from iPS cells when they are differentiated to more specialized cell types, including the development of assays for studying disease processes, scanning drug candidates for safety and effectiveness, or application to regenerative medicine. Comment: there is much older research by, and cited by Robert O. Becker, MD in his studies and books, of how the electrical polarity changes associated with injuries, as well as carefully simulated microelectrical stimulation, can activate a variety of adult cells to revert to a stem cell-like function of pluripotency to repair damage. He was an orthopedic surgeon and researcher. Most of his work was published in the 1980s. We invite you to learn more about our innovative approaches to helping you optimize your health. Check out our website at ARTC Health, subscribe to our newsletter to receive future updates, or call us to schedule a consultation. ARTC Health, 941-806-5511, doc@artc.health.